Hey everybody, welcome to the Intertidal lab. And this is a great lab because we get to go out into the field and look at how actual marine animals are adapted to the environment. Now we are going to be focusing on the rocky intertidal zone. Now there's actually not a lot of rocky intertidal zones here in South Florida. Wonder why? Can you come up with a reason? I will tell you this, there are a few examples of it. And we're going to check it out right now in the field at a place called Coral Cove in Jupiter, Florida. So let's go. Here at Coral Cove in Jupiter, the Anastasia Limestone Formation is exposed right along the seashore, creating a rocky intertidal zone. This sandy coquina limestone is a ratio of quartzite sand and shell fragments. These fragments are held together by a calcareous mixture. Just offshore, this same limestone provides nearshore reef structures for many fish and invertebrates. See if you can recognize any of the fish we've already learned about this year. Now let's turn our focus back to the intertidal zone itself, where a range of unique organisms make a living in a rather dynamic habitat. Think about what kind of extreme environmental conditions animals might experience living between the low and high tide watermark here on these rocky outcroppings. Now that you've gotten a chance to see the intertidal zone that we're going to be working within, I want you to think of the different environmental conditions that the marine life here might face during the course of a day or even a year. Make sure to list them out in your intertidal lab assignment. So now it's time to actually check out some of the marine invertebrates we are going to be tracking here in the intertidal lab. And I've got a few of them right here in these watch bowls to check out. Actually, the entire intertidal zone is named by this first organism who's actually trying to crawl out of the little watch bowl, and that is the Littorina snail. You might have heard the littoral zone. So let's get a little close-up on some of these organisms. These little Littorina snails are great at adapting to intertidal life. You can tell here in the watch bowl, they're actually already climbing up the sides and along the rim. Now out in the rocky intertidal zone itself, they'll often be exposed during low tide, and you can see them here clustered together. This strange creature is called a hairy chiton, and it's a mollusk, essentially a type of snail, and it has an 8-segmented shell that you can kind of see in the middle, and it gets the name hairy because of that fuzzy algae stuff all the way around it. Now, if you look underneath of it, it actually has a muscular foot, and you can see it, that's that orangey flesh down there, and it uses it to attach to the rocks and really hold on tight when the waves come up and down. It's an algae eater, so it scrapes algae off the rock for food. These two animals are limpets, and they are a type of mollusk, so a type of snail, and they have low-profile shells that help them manage wave stress in the environment. But they also have the ability to really seal themselves onto the rocks so that as low tide comes around, they don't lose a lot of water. They keep it sealed within. Now here you see the whole muscular foot on the back side of the animal, and that helps that limpet move around on the rocks and of course graze algae. The last animal I want to include is the barnacle, and of course I can't take that off the rocks. So here is a picture, and you can see some chitons in this picture, but also you can see the barnacles. And barnacles are really neat because they are arthropods. They are essentially related to crabs, but they're a really interesting morphology that is sessile, meaning that they are fixed permanently on the rocks after they settle from their larval planktonic forms. Barnacles are filter feeders, which means they feed on plankton as they drift by. Now, along the rocks, you're probably going to see this kind of honeycomb sort of structure. These are actually built by worms, and if you get close to them, you'll see that there's individual holes, and each of those individual holes is from a specific worm. If you get even closer, you'll notice that those kind of tubes are constructed of little pieces of sand grains. These worms, which are related to earthworms, actually take sand out of the water column and glue them together and create these tubes, and they do it in a colonial fashion. The neat thing is underwater, they actually create these mushroom-like structures that provide additional reef structure for marine life, which is really neat to see. These worms are actually filter feeders, so as the water comes in and out, they're actively feeding on plankton. Now that you have a bit of a visual on some of the animals we're going to be studying in the intertidal zone, take a moment to identify them on your lab assignment. That way you know what they are when you start doing the data collection in just a little bit. Also, think about some of the different adaptations you learned about in your notes for organisms in the intertidal zone. See if you can list out an adaptation for each of the different organisms we just looked at on your lab assignment. Now it's time to start designing the experiment. We know quite a few things now. We know the different invertebrates that live here on this intertidal zone. And we also know that we have a vertical rock face that we are going to be investigating. Now, the question then becomes, how do these different invertebrates distribute themselves along the vertical rock face that we've been studying? Now, we need to identify our variables. What would be our independent variable for this experiment, and what would be our dependent variable? Remember, the independent variable is what we are manipulating, and the dependent variable is we're measuring to see if it responds to that independent variable, right? So again, remember, we're looking at the distribution of these organisms along a vertical rock face from essentially where the water is, the low tide mark, up until the upper high tide mark, higher on the rock face. Now, with those two variables identified, we should be able to write a hypothesis statement. Now remember, you can easily use the if, then, because hypothesis statement format that we learned earlier in the year. So if you change or manipulate the independent variable, then what is going to happen to that dependent variable? What are you predicting? And of course, why do you predict that? One other thing we need to take care of is the control/confounding variables in the experiment. What are the variables we want to try and minimize change in to make sure that they don't influence whatever might happen in the dependent variable? Now in the lab, this is a lot more specific because we can literally control those variables. We can make sure the temperature stays the same, we can make sure lighting stays the same, that kind of a thing. But out in the field, we don't have as much control over those things, but we can monitor them. So again, on your lab assignment, go ahead and fill in all of these different components. The independent and dependent variable, what you think your hypothesis is, and of course, the control/confounding variables and how you are going to monitor them in the field. It's time to start getting into the details of how to run this experiment. Now, before we get too far involved, we want to always keep in mind safety and ethical considerations for any experiment. Whether it's in the lab or in the field, you want to make sure that you stay safe during the process. And for the field, we want to make sure it's safe to be out where we are going to be collecting data. So this might be as simple as checking out the weather report to make sure there's no thunderstorms, making sure you're able to access the environment. So in this particular instance, if I went at high tide, these rocks would be a little bit harder to access, especially safely with waves coming in and out. So I made sure to look at the tide charts and find low tide. So we had mostly an exposure of all the rocks to be able to get out there. And of course, I made sure that it wasn't thunderstorming, there was no inclement weather, it was safe to be there. Not to mention, it is a lifeguarded beach, so there are safety parameters around there. Now, in terms of ethical considerations, we want to make sure that we're not doing any harm to the environment. So I'm not necessarily walking on the rocks, I am not manipulating any animals in a harmful way. But because I am doing work with these animals, I do have a permit that allows me to go out and collect this information and manipulate and collect some of the animals for observations, like I did here for you guys today. And I can release those animals back to the site that we collected them from, which is great too. This permit comes from Florida Fish and Wildlife, and it's an educator permit that allows me to do some of this work. All I have to do is notify the authorities that I'm going out that day. And it's really important to make sure that you know the laws and regulations that are surrounding whatever types of field work you might be doing in your local area. Now that we've got the safety and ethical stuff out of the way, let's talk a little bit about how we're going to run this experiment. Now first, the classic and traditional way to do this is tried and true. Is a transect line, and we use a meter tape. This happens to be a 50-meter tape, which is way more than we need. And you can see it is measured in meters and centimeters here on one side, and then of course inches and feet on the other. We are scientists, and we are going to be using the metric system, so we're using meter side. And essentially what you do is take this line and let it drop across the vertical rock face. Thus, we are able to document the abundance and distribution across this line. I also have a dive weight for my free diving weight belt that we can attach this into, so it kind of helps it from flopping around in the surf too much. Then we come down to how we're actually, what areas are we going to actually count, right? So generally we use what's called a quadrat, and this is an example of a quadrat. It's a 10 centimeter by 10 centimeter square creation that's incredibly high tech. Just go to your local home improvement store and get some PVC and some 90 degree angle there and joints and put them together. You can do them in any sizes you want. This happens to be a 10 centimeter by 10 centimeter square quadrat. Then we've got the big enchilada, full meter. The full meter quadrat, which is pretty big. And this is 100 meters, or 100 centimeters, I should say, all the way across, right? Each square. Then I have the half meter. Here's the half meter square, 50 centimeters square, like that. Now, what size of quadrat should we use for this experiment? Let's throw that question out there for a minute and consider what it is that we're actually measuring. Consider the organisms we just viewed and identified. How big are they? How many of them would be in a half meter or a meter square? How many would be in a 10 centimeter square? Which makes the most sense to you for this style and this particular experiment? Think about that for a second. Now, if you are coming to the conclusion that the 10 centimeter square is probably better off, you're probably right. Our intertidal zone is only a couple of meters high. It's relatively narrow considering other intertidal zones at more active plate boundaries. So we're not going to be doing a huge distance here. And the small nature of some of our marine organisms are kind of going to be prohibitive. Even in a half meter square like this, you're probably going to be counting thousands of littorinus snails in some parts of the intertidal zone. So the 10 centimeter by 10 centimeter square is probably going to be better for our purposes. Now, traditionally, in some of the more diverse and kind of larger intertidal zones, like in the Pacific Northwest, half meter squares are very typical for that environment, just because some of the animals are a little larger and more spread apart, that kind of a thing. So that gives you an idea of some of the equipment we are going to be using. So we have one other consideration to talk about, and that's random sampling. Now, random sampling is a really important part of any sort of scientific experimentation, especially in the field, because we want to make sure that we are not including bias in our data. What does that mean? Let's say I believe that chitons are going to be found in the upper intertidal zone. Well, I may even subconsciously pick a line, a transect line across a rock face where I just happen to see more chitons. To avoid that and any semblance of bias, we want to not pick our transects just because we want to do it there, right? And so one of the ways we can do this, and I'll show you an image here on the side, is that I can do sort of a horizontal line, transect line if you will, and essentially generate a bunch of numbers. And there is something called random number generators that you can go out online and find. There's plenty of apps that do it. And you can essentially have them, and you can do this before you even get to the field, that way you just have them aside. And you put your horizontal transect line down there across the top, and then you select, in this case, we're going to do 6 different transects, 5 or 6, depending on your lab you're doing, but most likely 6. And so out of those, you maybe have 10 different options, and you do a random number generator, and it gives you 6 numbers, and that would be the location of your 6 vertical transects. So you didn't specifically pick them. The random number generator picked the location on the rock face that you're going to do and carry out your work. And that way, you know you're not being biased. You're not seeing things and just subconsciously like picking things because they match your hypothesis or your prediction. That's a really important part of dealing with scientific data collection. All right, so now it's time to get out in the field. So let's look and see what this looks like with me and Sarah out there trying to collect some of these images and some of these transect data. Here we are at Coral Cove, walking out to the rock face to start our transect lines. Now, truth be told, I actually took a lot of this transect data about a couple years ago, designing a lab just like this, but I wanted to get some fresh data for this year. However, the water was a bit rough even at low tide, and you just never know when you're sitting in the field what it's going to be like. But we gave it a good shot. And then, of course, here comes a wave. And that is me running to make sure my camera that is filming does not get washed into the ocean. I can assure you that all equipment and personnel came back from the field safe and sound, no problems whatsoever. But now it's of course time to turn it over to you to do some data collection on what we actually are providing. So you have access to a series of PDF files that have the images of each of the different quadrats labeled within each transect. And remember, this is a replication situation. Replication is hugely important. That's why we're doing 6, right? 5 or 6, because we want to make sure that we have a good sense of what's happening on this intertidal zone without any particular bias. If you only did one transect, maybe there's an anomaly in one or another area. So by doing more transects, you're hoping to kind of alleviate that and make your data more reliable over time. And so we're going to be working with that. We're going to collect means of these different transects at the different levels of the intertidal zone, and you're going to be graphing that stuff. So you want to pay attention to in-class instructions as to how you're going to do this, because you're going to kind of access it as groups, right? So each of you will have different responsibilities, and that might change from year to year. So you'll be accessing these, blowing that PDF image up and counting the organisms there and inputting that information into the data sheets. So I hope you enjoy kind of going through this a little bit. It's actually kind of fun, I think, because when you get to the end, you get to draw real world conclusions. And of course, the very end of the assignment has some of those style questions for you to essentially contemplate what's going on between this species and that species and their position in the intertidal zone. That's the fun part of science, I think, was when you get to unveil things that maybe people didn't know before. And that's what you're doing today. Thanks for joining us in the intertidal zone. Keep learning. Okay.